Welcome back, everyone, to the 2021 NDSU Soybean Symposium. Our first speaker this afternoon is our host, Dr. Carrie Miranda. Dr. Miranda earned her Bachelor's of Science in Biology from Cleveland State University, her Master's in Molecular Biology from San Diego State University, and her PhD in Plant Breeding and Genetics from the University of Missouri. During that time, she was awarded a Borlaug Fellowship to conduct her dissertation research in Ghana, West Africa. She worked as a private company pea breeder and a postdoc at the USDA before joining NDSU as the soybean breeder. Dr. Miranda, I can see your presentation. I'll send it off to you.
Thanks, Cammie. And I also wanted to say thank you, Cammie, for all the excellent work that you've done to help set up this presentation, along with Scott Swanson as well. Extremely grateful, and I think today is going off really well. Oh, and I want to add too, take off my presenter hat, put on my MC hat again for a moment. All of these presentations are being recorded and they'll be cut so that if you just want to watch one person's presentation again, that you'll have the ability to do that. And those will be on the same website where you registered following the end of today. Okay, so now back to my speaker hat. Hi, everyone. I am Dr. Carrie Miranda. I'm the soybean breeder at NDSU. Relatively new. I started about 7 months ago. And my presentation, which should make the Soybean Council very happy, is Grain Cards Full of NDSU Soybean, the future of the soybean breeding program. And the reason I have this mentality is that at NDSU, the breeders here are in a very unique position that we have a lot of infrastructure and a lot of support to not only create varieties that can be released through foundation seed stocks, but then they're promoted through the crop improvement lab as well. And there are a very good percentage of North Dakota farmers that grow NDSU varieties of all 10 of our different crops that we produce here. So I am highly motivated to release varieties. And my hope is that I can produce so much amazing NDSU soybean that Corteva and Bayer, a thing of the past. No one's even going to consider them anymore because they're going to be like, oh, wow, public varieties are so fantastic. So before I get started, I would like to give credit to Ted Helms. Ted Helms was the soybean breeder before me. He retired last summer. He was the soybean breeder at NDSU for 34 years. So he earned his Bachelor's of Science from the University of Illinois, his Master's at the University of Nebraska, and then completed his PhD at Iowa State University. Very similar to my mentality, he was very variety release motivated, and he released 40 soybean varieties throughout his career, which means that he was able to release more than one variety for every year that he worked. But most importantly, he said that he loved teaching and is quoted as saying, teaching was always one of my favorite parts of my job responsibilities. So Ted was a really excellent breeder. He set up this program for success. I'm extremely lucky and grateful for this. And I know I have some big shoes to fill. Okay, so speaking about the soybean program, I see it in two different ways. One aspect is breeding for variety release, but then also doing the breeding research so that we can find those high value traits, and then also the agronomic soybean cultivar improvement. So I'm going to go through breeding for variety release first. And this part, I am completely dependent, reliant, and grateful for my technician team. My head technician is Ashley Cooper. She's been the head technician for the past year, but has been with the soybean program for several years. Aaron Fromke is a technician and who has a master's in weed science. So obviously his expertise for selecting chemicals and managing our weed control, most public breeders will tell you is one of the biggest challenges. A farmer will tell you that too. Weed control is a huge challenge. And so I really appreciate his expertise. And then as we collect more resources and have quote unquote more time, we're going to start doing some weed science research as well. Brock Fagerstrom has been with the soybean project now for 7 or 8 years. So he has not only historical expertise, because I can ask him, hey, what happened 5 years ago? Why is the data off? And, I can know what the weather was like then. But he's also really excellent at repairing a lot of our large equipment. And then Jerry Gee is a longtime technician as well, and in a very positive way. Jerry is retiring this year. So we're excited that he's been able to finish his career in a good way with NDSU. And we are currently in the process of hiring a new technician who will be assuming a lot of molecular work. So whenever you hear me talk about molecular markers in the future, in this presentation, that's what the future technician will be doing. Okay, so at this moment, there are 4 main breeding projects that I'm focused on: conventional and food grade, high oleic, glyphosate tolerance, and then nutrient deficiency and disease tolerances. Breeding for, okay, so conventional simply means that there are no genetically modified traits or genetically engineered traits in the soybean. Currently, the conventional program contains the highest yielding line. And this is a pamphlet for one of the marketing pamphlets for an NDSU variety, ND Benson. And with the dependence of farmers on glyphosate, or excuse me, on herbicide tolerance, conventional soybean has become rather niche. And one aspect of that is because of hilum color. The hilum is simply where the seed connects to the pod, but you can see that there's a wide variety of colors associated to the hilum. And ND Benson has what's called a light brown or buff hilum color. And you wouldn't think that that would be significant, but it actually is. So when it comes to conventional soybean, this yellow, white, clear, there are a lot of names for the same phenotype. This yellow hilum is much more advantageous. And let me tell you why. Because a lot of the conventional soybean now is actually used for soy food products. So if you look at this top picture of tofu and soy milk, it's very white. And if you look at the bottom picture, that it has a gray tinge. Now, this is an exaggeration. You won't get this dark of a gray color from brown hilum soybean. This is actually made from black seed coat soybean. But you can see that even that small pigment can still have an impact on the purity, the whiteness of the soy food, and there is not a market for that currently. So in my mind, I think that it's necessary to convert a lot of my conventional soybean to this food grade market. So what are the components that go into food grade? I'm going to talk here specifically about tofu. One, it's large seeded. As I said, it has the yellow hilum. It's high protein, 40% or more, high sucrose. And there's also this trait called low lipoxygenase, which is an enzyme that when it's not present, you don't taste that beany flavor that is associated with soybean and other legumes. And so all of these traits together create what I call a high value traded soybean. So what's great about this is that there are markers available for a lot of these traits, many of which were developed by Kristen Ballou, who's our keynote speaker. And you don't need to have all 5 of these. You don't need to have all 5 of these traits in order to have value in your soybean. However, you have more value the more traits that you stack together. So even if you have a population that's segregating for these traits, all of them have a market someplace. And so that's the direction that I would like to go through for tofu. Another soy food is natto. This time it's the other direction with seed size. Now you want as small as possible. Here, protein isn't as important, but you still want the high sucrose, and that in getting rid of the beany taste is, I don't want to say controversial, but it's age dependent. I spoke to one of the food grade producers and they said older people like the beany taste in natto and younger people don't. So it's an ever, this is a consumer-based market. And so you have to anticipate what people will eventually want, which is kind of fun. Okay, another aspect of my program is high oleic soybean. It was already mentioned earlier by Kendall, but I'm going to take you through the history of why high oleic is needed, because I think it's a really interesting story. So soybean oil on its own is unstable at room temperature and high cooking temperatures. So in the 1900s, they started putting soybean oil through this process called hydrogenation. And a hydrogenation is what it sounds like. You add hydrogen atoms, and it changes the chemistry of the fatty acid chains. So when you have complete chemical hydrogenation, then you have it breaks the double bonds. And so in complete chemical hydrogenation, you have actually a very, very stable molecule. There is nothing wrong with complete chemical hydrogenation. The difficulty from a consumer standpoint, though, is that fat is solid and it's not very useful for cooking purposes. And so consumers wanted something that was softer. So imagine, everyone can probably picture this on their grandma or their mother's counter back in the 80s, if you were alive then. They all had that blue can of Crisco shortening sitting on their counter. That is a perfect example of partially hydrogenated soybean oil because it's soft. It's still soft at room temperature. However, when you do this, there are still double bonds in that fatty acid chain, which then change the chemical conformation. So instead of going from a typical cis conformation, now you have changed it to a trans one. So everyone remembers their chemistry, right? Cis and trans are just mirror images of the same molecule. And so you would think, what's the harm in that? But there's actually a lot of harm. Trans fats have, it was determined in 1993, although it had been suspected for decades earlier, that there is a negative effect of eating trans fats on heart health. And in fact, in 1994, it was discovered that 30,000 deaths per year were contributed to heart disease caused by consuming trans fats. And 20 years later, I don't know why it took that long, or 10 years later, in 2006, the FDA finally acknowledged that they need to be labeled. By 2013, trans fats were deemed no longer considered safe, which is the same labeling as an actual poison. And by 2015, trans fats are starting to be phased out of food production. But at that point, it didn't matter. The damage was done. The soybean industry took a huge hit, and that void that was created by removing soybean oil from the market was filled by palm oil. And by 2008, palm oil became the leading edible oil produced. Now, I know I shouldn't say this as a soybean breeder. Palm oil is delicious. However, it's extremely terrible for your health. It's really high in saturated fat and is, I believe, the leading cause of deforestation in Southeast Asia. So whenever you see those ads about orangutans losing their habitat, it's because of this soybean oil controversy. So I think this story is interesting because so many things that we've experienced in our own lives are actually affected by soybean in agriculture. So Kristen Ballou, and don't worry, I know she's the keynote, but I'm not ruining her speech. She's not talking about this one. So Kristen Ballou had the idea of, hey, let's look at the biochemical pathway for fatty acid synthesis. And at this point, oleic acid is stable at room temperature. However, when you keep putting it through the processing pathway and get down to linolenic acid, this is when the molecules become unstable. And so she had the idea of, if we mutate BAD2, we should see an accumulation of oleic acid, which should be stable at room temperature. And in 2010, she published this research. And for those of you that aren't so familiar with soybean genetics, it happens very often because soybean underwent a genome duplication millions of years ago, that if you try to delete one gene, which a lot of people had tried to delete that FAD2 gene, but they didn't see the elevated levels of oleic acid. But then a lot of the times you have to think, oh, there was a genome duplication. There's probably a second copy of this gene that's still functioning and making up for the deletion that I caused in the first copy of the gene. And she was absolutely right. This graph is showing a percent of oleic acid, and she was able to find 2 mutations, one in each copy of the FAD2 gene. And you can see here that parent one that only has 1 gene mutated only has about 28% oleic acid. The second parent, which has the second deletion in the second copy of FAD2, has about 41%, but that's still too low to be commercially and economically stable. However, when you combine both of those mutations together, that's when you get that 80% oleic acid, which is necessary for commercial production. And so that is the long story about why we're bringing high oleic into the soybean program. It's a healthier oil than palm oil. It's more economically sound, and it has a premium. It's a high value crop for North Dakota farmers. Next is our herbicide tolerance program. So currently we are working with glyphosate tolerance, which has been very successful. And I hope to bring in other types of herbicide tolerance into the program as well. So here I'm showing the pamphlet for the marketing materials for a very, very successful line, ND17009. It's a 0.9 line. And farmers in North Dakota find a lot of value in this because it's a low cost herbicide resistance, and farmers can also save their seed. So there's a lot of contractual obligations when you buy new lines from private industry that you cannot save your seed and plant it again the next year. But NDSU, since this glyphosate tolerance gene that we utilize is off patent, we don't have those same licensing constrictions. And farmers do really value that. And so for this program, I want to continue adding high value traits and increase yield. So this is the program where I'm very motivated to increase yield as quickly as possible. So I went to the Soybean Breeders Workshop in February, and someone there mentioned that HighThoroughput Phenotyping is a more accurate prediction model for yield than genomic prediction. And it totally blew my mind. And I'm very interested in testing this out because as far as I understand, it's a very new idea and has not been tested in the soybean model yet. My hypothesis is that it's going to be a combination of both genomic prediction and high throughput phenotyping modeling that's going to give you the most accurate results, but then we have to work to, make it cost effective as well. So implementing the new technologies into my herbicide tolerance project is going to start happening this summer. And of course, what's a breeding program without protecting those yields that we're fighting so hard to get? So these 3 challenges are something that I'm very focused on. And thankfully, a lot of the previous speakers set me up for this. Iron deficiency chlorosis is, as you can see, chlorosis of the soybean caused by alkaline soils that don't allow iron to become available to the plant. This is a challenge in the Red River Valley, which if you remember, Hans showed that map, is in the eastern part of the state and the border of Minnesota, which is also where a lot of the high value soybean is grown. So it's definitely in my interest and the farmers' interest of North Dakota to ensure that our highest yielding varieties also have the most excellent stringent form of IDC tolerance possible. As we already learned, soybean cyst nematode is a problem. And if we mirror the history of the other Midwestern states, it will become a very big problem. As I mentioned before, if you heard me during Yuping's talk, I mentioned that since since soybean production in North Dakota started a few decades after the rest of the country that produces soybean, we can kind of learn from what they've gone through, which also includes which forms of SCN resistance are we going to utilize in this program. And so that is also a extremely important, I cannot stress that enough, extremely important aspect of this program. And then we can't forget our favorite, Phytophthora root rot. Phytophthora root rot is a stress in the state. And although we do have good sources of resistance to it, it doesn't mean that we can neglect it in favor of only researching IDC or only researching SCN. It's not like Phytophthora is going anywhere soon. And so this breeding program has to be a solid balance of incorporating all these resistances in order to protect those yields, those gains that we've worked so hard to create. Fortunately, a lot of this can be done with molecular marker use, which I will be implementing into the program. And now going into breeding research. A lot of my past research has been in maturity. And if you worked in soybean for a while, I'm sure you've seen this maturity group map before. So North Dakota is able to grow double zeros, zeros in the very southern part, group ones. Okay. And Kristen Blue, again, and Tiffany Language, her postdoc, did some really excellent work genotyping the E genes to understand their allelic combination control on maturity groups throughout the United States. And it was found that a lot of the E gene alleles that are used this far north are non-functional. And so I believe that minor effect genes, like the flowering time genes that are characterized in Arabidopsis, or even other smaller effect genes that we haven't discovered yet, are what's controlling flowering time in North Dakota. Or from what I heard from Anna Dokes and what I've seen in the short time that I've been here, it seems like soybean in North Dakota is influenced by growing degree units, which kind of blows my mind because when I was in Missouri, flowering time and maturity are almost 100% heritable. And so I'm really interested in conducting an experiment to understand what is the environmental effect on maturity in North Dakota soybean, because once we understand that the fine tuning of maturity for the specific environment, then we can start playing around with stem architecture, which Junghwa Kim in the student presentations did a really excellent job of showing how these different alleles of the DT1 gene, which controls stem determinancy, that these allelic changes can actually have potential impact on yield. And so the way I envision it, I would like a short season crop that still can put on as much yield as something that takes 130 days. So, historically, and Hans talked about it in his presentation as well, farmers are always fighting to get that later maturing line because that usually leads to yield gains. But what if we can control the season length to be exactly what we want to be, and then make up for the shorter days by influencing how many pods we put on the stem? And after that frost we got, what was it, September 10th of last year, I am very motivated to ensure that North Dakota farmers have a short season soybean that's still high yielding, because we want to risk, frost risk is a severe threat here in North Dakota. So going on from that, I also am very interested in comparative genomics. I like to call us the genomic dream team. I'm the soybean breeder. Phil McLean on the left is a bioinformaticist who did a lot of really excellent IDC work. I actually got my markers from some of his publications. And now he does a lot of bioinformatics work with Juan Arsono, who's in the middle picture. He's a phaseolus breeder. And then Ninoy Bandillo is a newer faculty member here at NDSU who has worked in soybean during his postdoc and is now the pea breeder here. So he and I kind of flipped because I was a pea breeder and then became the soybean breeder. And so I'm really, since soybean is so rich in genomic resources, I'm really interested to leverage that to see if the same gains that we found in soybean can be translated to phaseolus and then to pisum as well, which is now becoming a newer cash crop. And again, maturity and stem architecture are my focuses there. And then finally, I know I'm running short on time, but I'm also the MC, so there's no one to yell at me. So finally, Cammie mentioned in my introduction that I did my PhD research in Ghana in West Africa. And this is one of my favorite photos of my entire life. This is a photo of Nicholas Denwar on the left. He's the soybean breeder of Ghana. In the blue shirt is Andrew Skibu, the soybean breeder of Missouri. And then on the right is Brian Deers, the soybean breeder of Illinois. We were all part, and still are, a part of a USAID project called Soybean Innovation Lab. So they were there in Ghana facilitating breeding improvements on a higher level, whereas I was there doing my yield trials. But these three breeders really became excellent mentors to me, and they walked my fields and gave me their opinions. And certainly that's when I felt like, oh, I'm really a breeder. And then this photo was taken at a national park in Ghana, in between looking at fields, actually. So I published this research this is my main maturity work, dissertation research. And very quickly, I'll go through this. If you take an American soybean variety and grow it in close to the equator, so low latitude, you have short days, 12 hours of daylight. What happens is that short daylight sends a cue to the plant as soon as it emerges to start flowering and then start maturing. And so that's why you have this tiny, tiny little brown early maturing line that has only a few seeds on it. You can see the impact on yield it has. Whereas if you utilize something called the long juvenile trait, it tells the plant, hey, don't worry about those 12 hours of light, keep growing green. And then you see an increase in terminal stem height, which allows the formation of more nodes, more pods, and higher yields. The conventional variety grown in a 12-hour short day will mature in about 90 days, whereas you would see longer season with a long juvenile trait, you can control it, but say 120, 130 days. So the long juvenile trait was cloned in a gene called ALF3, an Arabidopsis ortholog. And so when we developed our populations, we looked for parents. So these are the parents that we chose based on their ALF3 allele. So 3 of the parents were conventional, and then we crossed them by lines that we knew had the long juvenile trait. So PI 159925 is historically where the long juvenile trait was first found. That has a deletion in ALF3. Parana-Guyana, and then this Canadian experimental line, which derives its long juvenile trait from Parana-Guyana, also have a mutation in ALF3 that gives you the phenotype, that gives you the long juvenile phenotype. Then we also used a Ghanaian variety called Janguma, but when we sequenced ALF3 in Janguma, we did not see any causative mutations in there, which means that there's probably a different source of the long juvenile trait or delayed flowering in short days in this line. And also I wanted to point out that we also took stem architecture into consideration in this experiment because, once again, I really want to drive home that message that the correlation between maturity and stem architecture has a tremendous influence on yield. So this figure is from the supplementary of that paper, which is where all the good data always is. And I'm not going to go through all of it, but let's look at this box that's highlighted in green. These are histograms of days to maturity. So the days to maturity are, no. The days to maturity are on the bottom, and then the number of lines are on the y-axis. The arrows pointing down are the days to flowering for the parents. So the conventional parents is on the left, and the long juvenile parent is on the right. So in this first population, X97 is conventional. This was crossed by PI 159925. And I should add that in the lines that were conventional, most of them didn't survive in this environment. I mean, you saw that tiny phenotype. They didn't really stand a chance. So you see here, the lines that survived all had the parental phenotype, but I would assume that if the conventional ones would have successfully survived, that we would see a bimodal distribution of days to maturity, early and late. But when you look at the X97 by Janguma population, that's not what you see. It is not bimodal. In fact, you have a normal distribution of days to maturity in between the two parents. And I wish there was a grad student here that I could pick on and say, what does that tell you? But I'll give you the answer. So that tells me that there is that the long juvenile trait in this Janguma population, or in Janguma, is a quantitative trait. That it's not a single gene, unfortunately. I prefer the single genes, of course. And so the next step of my research is to understand what gene combinations are creating this phenotype. And I have a master's student coming in from Nigeria who's very interested in this research and hopes to continue breeding in Africa. So I'm really excited to be able to continue this. Then also, I'm like, where did this African soybean even come from? There is not, it's a novel source of the long juvenile trait is what I'll say. So I want to say I did it. I'm pretty sure I just had the idea, and then I absolutely could not figure out the R script to create this PCA. So this is a PCA analysis using GBS data of African lines. So the phenotype data that I had for the African lines was extremely limited. I didn't have any phenotype data, and I didn't even have country data. So these, I don't really have time to explain a PCA analysis if you're not familiar with it, but these blue dots are the African lines, and then the red dots are from China. So not surprising to see that most lines are overlapping with China. And then you have soja, because I thought maybe soja had a role in African germplasm development, but apparently not. So what we did, we had GBS data, and I asked, I'm sure I did not do this by myself. I'm pretty sure I asked Nick Dietz. So that's his photo here. Nick Dietz, he was a fellow graduate student with me. He is an excellent bioinformaticist. He also happens to be graduating. So if you need a bioinformaticist postdoc, he is the person to hire. It's fantastic. And so we extracted the GBS SNPs from whole genome data of these lines where we knew that we had country data for. And that's how we were able to construct this PCA analysis. And it's just interesting because the African lines don't associate with much of anything except a few in China. And then, of course, they don't even necessarily associate with themselves. And since I don't have the country data from where these lines came from, I'm going to assume that that this is probably a West versus East Africa. But I need to do more research to determine that. And fortunately, now there are more genomic resources for these African lines, plus more data. So I think that will facilitate understanding where did African germplasm come from. So in summary, I'm breeding for variety release, high yielding, high value traits. And then my breeding research focuses in maturity, stem architecture, and international response to improve food security. Wait, let's see if it works here. Yeah. All right. And with that, I'll take any questions. Okay, I see a question from Yiping Yan. What major breeding methods would be efficient for breeding conventional soybeans? And what major breeding methods would be efficient for breeding GMO? So I don't necessarily see there being a difference in breeding methodology between conventional and GMO. What I see are the different breeding methodologies for what traits I'm breeding for. A lot of the qualitative traits I would select for early, essentially, which would include the herbicide traits. So, but like what's interesting for natto, yield isn't important. So I can make selections to increase lines way, way sooner because I don't need 3 years of yield data like I do for all these other lines. So I definitely think it has to do on what the specific project is that I'm working on. And then Kristen writes, will you do whole genome assessment of your germplasm? So this is the question I've been asking Aaron Lorenz, Andrew Skaboo. How do you start incorporating genotyping into a full running program, right? Where do you start? And so that's why I was saying that I think that starting with the glyphosate program is most prudent because then I could immediately start using those for genomic prediction. So I don't think that I would start any if you're referring to whole genome sequencing or even skim sequencing, I don't think I have a need for that yet. If anything, I would want to assess specific traits first because I don't know what maturity genes we're currently using in this program. I know some disease resistances that we use just based on the parents that were donors. Oh, sorry, I was distracted by a question that was asked. And so, but when it comes to the research side, if there are lines of interest to whole genome sequence, I will. But I would like to have a marker panel that could tell me all the trait information that I need across the majority of my program. That is the ultimate goal once I learn how to organize and manage all that data. Okay, there's another question. Does that 40% protein of your large C line dry matter or 13% moisture? I'm not 100% sure. I'm pretty sure it's 13% moisture, but it's really the consumers of that who make those decisions. Really, I'm just like, oh yeah, because I would take the protein percentage with moisture. So, and then I give it to them to evaluate further.